Know that numbers of different nutrients are required to organisms. Is there any system where we classify the organism on basis of nutrients requirement? Can we say that this particular organism requires this particular things, or can we classify them systematically on the basis of their requirement? Then the answer is yes. We have a way where we can classify the organism systematically in form of nutritional requirement of them. The first and prime criteria for this classification, that is on the basis of the energy requirement. The energy is crucial for all the living creature. Similarly for the microorganism also. The energy is required for various anabolic activities, where it goes for synthesis of protein, or it goes for replication of its DNA, it goes for transcription and formation of its RNA, various types of RNA. So major cell constituent to be formed where the energy is required. And this energy can be utilized from various substrates. Now, on the basis of energy requirement, we can classify the organism, the first type, that is called phototrophs. Phototrophs are the kind of organism which can utilize the radiant energy in form of solar radiation. The photon energy which can be utilized, the organism do possess a photosystem which has got capacity to absorb the kind of photon energy and convert this energy into chemical energy. And the excellent example, that is the cyanobacteria, which has been projected in front of you, that says different cyanobacteria varieties are there which utilize light energy and grow. Another type, that is chemotrophs. The other than phototrophs is chemotrophs, where these are the organisms which rely upon chemicals as a source of energy. Organism which can utilize chemical compounds, maybe a kind of carbohydrate or maybe a other kind of fat, can be degraded by the organism and energy is derived from that. All these organisms comes under the title of chemotrophs. The best example is your routine organism, Escherichia coli, or the Bacillus species, which day to day in microbial laboratory we used to grow. All these organisms are kind of chemotrophs. Another criteria for classification of organism, that is the source of electrons. Means electrons donor is very important. Here there are two types, that is lithotrophs are the kind of organism which can utilize reduced inorganic compounds as electron donors, and they are termed as lithotrophs. Another one is organotrophs, where the organism which can use organic compound as electron donors, and here the electron supply comes from the organic material. So litho and organo are the two types on the basis of the electron donors, and photo and chemo are the two types where on the basis of the requirement of the energy. Where you go for photolithotrophs, here two things are combined, that is the source of energy as well as source of electron donor. Here photolithotrophs are kind of organism which are phototrophic, so they rely upon radiant energy, and at the same time they can use inorganic compounds as their source of electrons. Say for example H2S can be utilized by this organism, as we have mentioned that these are the organisms which has got tremendous capacity to use a kind of inorganic sulfur, and H2S is converted into sulfur, and elemental sulfur can be cleaved off and electrons are being utilized. Chromatium ocani, that is the kind of organism which has been projected, that is utilizing the energy from H2S, that is a kind of photolithotrophs. Another one is photoorganotrophs. Here energy that comes from the radiant energy, that is photo, and the electron donor kind, that is the organic. Organic compounds, say for example succinate can donate the electron, so succinate itself convert into fumarate, and two of the electron that comes out and which can be used by the organism for its cellular function. The excellent example in this case, that is the Rhodospirillum rubrum, the organism which is projected, that is a spiral shaped flagellated bacteria which are using energy source as the light energy and electron donor as the organic substrate. Another one is CHEMOLITHOTROPHS. Here the energy that is relied upon the chemical other than radiant energy, and the electron donor that is kind of inorganic. Here CHEMOLITHOTROPHs are the organism which can utilize ammonium ion that is being used as electron donor, and ammonium is converted into nitrate and ultimately the electron is derived from that, and the organism is termed as CHEMOLITHOTROPHs, so organic chemicals. And best example is Nitrosomonas species, Legionella species, which is projected in front of you. These are the chemolithotrophic organism which has tremendous capacity to deal with ammonium ions. Organism utilizes it and converts into nitrate. All these organisms are farmer-friendly organisms. Another category is the chemoorganotrophs. Here chemotrophs meaning they rely upon the chemical energy. Another one is organotrophs meaning they are the organism which requires organic substrate to donate electron. And the best example here is the glucose, which is the excellent electron donor molecule, organic substrate. One molecule of glucose that yield 24 electron to the cell, and the normal organism which are routine in microbial laboratory, that is Pseudomonas, which can use the organic substrate as electron donor. So chemoorganotrophs kind of Pseudomonas, Pseudoflava that is projected in front of you, even other Pseudomonas are also the varieties, different species of Pseudomonas can utilize the chemical energy and the organic electron donor. The rest of the classification, Dr. Kiran Rajput will explain you.
Nutritional types of microbes on the basis of carbon requirements. So carbon is the most important requirement. Many organisms in nature are there. They can utilize carbon dioxide as a sole source of carbon for assimilation of various cell components. So these organisms are known as autotrophs. Heterotrophs are the other organisms which can utilize organic compounds as their sole source of carbon. And the third category is known as MIXOTROPHS. They can obtain energy from inorganic electron donors, but obtain their most of the carbon requirement from organic compounds. So these are the three major categories of on the basis of carbon requirements. Autotrophs are the organisms what we called as chemolithotrophic bacteria, which can utilize CO2, that is carbon dioxide, as a sole source of carbon. They are termed as autotrophs. Particularly Nitrosomonas, which is seen in the picture, is a chemolithotrophic bacteria or autotrophic organisms. These type of autotrophic organisms can utilize energy from reduced carbon sources like hydrogen, hydrogen sulfide, elemental sulfur as electron donors. Autotrophic bacteria can be cultivated in a simplest medium that we called as defined medium or synthetic medium. The requirement is very simple. Mixture of inorganic compounds, inorganic salt is supplied, and they have tremendous capacity for synthesis because they can fix the atmospheric nitrogen. Here the medium is shown for the growth of Nitrosomonas europaea. Here ammonium chloride, magnesium, potassium, calcium, manganese, sodium, molybdenum, zinc, copper is supplied, and organism will utilize major carbon requirement or a sole carbon from atmosphere. So organisms can fix carbon dioxide and then they will grow. So autotrophs can be grown easily. Next, heterotrophs. Heterotrophs are the bacteria which can utilize organic carbon compound as a source and as termed as heterotrophs. Heterotrophs are immediate concern to us. The greater population of microbial population in our immediate environment is of heterotrophs. For example, whatever the microorganisms which are of economic importance, they all are generally heterotrophs, as well as organisms which is producing various diseases in human beings, animals, plants. These are also heterotrophs. For example, here pictures of few heterotrophs is been shown, that is Escherichia coli, normal flora of our intestine, Azospirillum braziliensis, it's a normally present in soil, Salmonella typhi, which can cause disease typhoid, Proteus vulgaris, it is also a skin flora, Staphylococcus, skin flora, Lactobacillus acidophilus, which is a normal flora of curd. So these are the bacteria which are immediate concern to us. So normally heterotrophs are present in environment in ample amount, large amount, and they can be cultivated in two types of medium. Another heterotrophic bacteria, that is Burkholderia sepia. This is a most versatile organism which can utilize about more than 100 different types of carbon compounds. So these type of specific heterotrophs are there in nature which can utilize diverse range of compounds. Heterotrophs can be cultivated in defined medium as well as complex medium. Defined medium, as we have seen, it is having an exact composition, each and every component is known and weighed properly and then added in a medium. So here is the medium for growth of E. coli, defined medium for growth of E. coli. Next, complex medium, that is nutrient broth. Here natural components like beef extract, peptone is added in the medium. Exact composition of complex medium is not known, and that is why it is known as complex medium. Generally heterotrophs are grown in a complex medium. Next category, that is known as fastidious heterotrophs. Fastidious heterotrophs are the organisms which have specific nutrient requirement, that is specially vitamins and other growth promoting substance. In absence of this growth promoting substance, that is vitamins, these organisms cannot grow. So for growth of that particular fastidious heterotrophs, we have to supply this type of specific vitamins in the medium for growth of these organisms. For example, for cultivation of lactobacillus, we have to supply about 8 different types of vitamins in the medium for the growth of heterotrophs. So this is about the cultivation of heterotrophs. Now MIXOTROPHS. MIXOTROPHs are the organisms known as CHEMOLITHOTROPHIC heterotrophs, where they can utilize both type of substances. They can obtain their energy from inorganic compounds. So inorganic compounds will act as electron donors and will obtain energy from it. And as a carbon is obtained from normally organic compounds. So these organisms are known as MIXOTROPHs. Example is desulfovibrio desulfuricans as shown in picture. The other example is Beggiatoa. It's a green sulfur bacteria. It can also act as a MIXOTROPHs. Next will be explained by Dr. Pancha.
Another category, then all these organisms where we go for the cultivation in our laboratory, that is obligate parasites. When we say this organism, these are the organisms which cannot be grown on synthetic or laboratory medium. Actually, these organisms are causing disease to animals or human beings, and their nutritional and physical requirements are not been understood properly yet, and they cannot grow on artificial medium in the laboratory. Means that we are trying all our best to find the way to grow them, but it's quite difficult, and many a time it is quite impossible to grow some organisms in laboratory. We call them as obligate parasites, and if you want to grow them, then we need experimental animals like mice, where we'll have to inject the organism in the animal, and animal itself is serving as the medium for us, where the organism will grow there, and then after we can utilize them. The Mycobacterium leprae, that is an excellent example of the obligate parasite, where the organism causing leprosy. Normally, there is no way where we can cultivate it in laboratory medium. Well, if you need to grow them, then the mice, where we'll have to infect mice by this organism, and the organism is growing in the body of mice, and then after you can utilize it. That is the organism causing leprosy. Another one is Treponema pallidum, which is causing syphilis to us or to human being. This organism is a kind of spiral bacteria. Same in case of Treponema pallidum, where it is obligate parasite, cannot be grown on laboratory medium, but still try are going on where we try to cultivate them on experimental animals. Another one is Laptospira. This is a typical organism which requires long chain fatty acid as a source of carbon, and due to this character, the organism infect human being and the human cell which contain long chain fatty acid, where the organism can grow very well and cause disease to human being. The Leptospira is again dangerous organism. To grow them in laboratory, again it is quite difficult. Another one is Rickettsia, where the Rickettsia, again they are causing infection to human being. The projected slides shows some of the Rickettsia, which are kind of gram-negative, short, raw type organisms, which has got tremendous capacity to cause infection to human being. Again, growth is very difficult, but still the trials are going on where we can use blood, where RBC can be utilized as a source of nutrients to them, and the organism is growing on blood agar medium at some extent, but the prolonged growth that is not been obtained. So it's a kind of obligate parasite. Another one is chlamydia. Same in case of chlamydia also, that is human pathogen, can cause sexually transmitted disease, as well as they are the organism which are having the efficiency to infect, and it can cause blindness to human being. Same organism cannot be cultivated on laboratory medium. We use experimental animal for that, and the trials are going on to find a way how we can cultivate this kind of chlamydia on laboratory media, but still it's quite difficult to grow them on laboratory media. So obligate parasites are the organism which cause disease to us, and the growth on laboratory media is quite impossible. Rather, we do not understand its nutritional requirement as well as physical requirement, and some cases where the nutritional requirement is defined and understood, but the physical requirement cannot be provided properly. Hence, the organisms are difficult to grow on laboratory media, where we'll have to rely upon the experimental animals, where we'll have to infect animals, and animal can be sacrificed for the growth of such organisms. With all this, the nutritional requirement, the references as we have referred for this, that is normal microbiology title, that is 5th edition of Pulsar, Chan and Craig. Another one is Prescott, that's again title Microbiology, 5th edition, that is Prescott Hurley. Third one is the title Microbial Word, again 5th edition of Steiner, Edberg. And the last one is the Brock Biology of Microorganisms, that is by Medigam Mamti, Meritko, and the Parker J. All this reference, I would request Mr. R.J. Patel to sum up the talk.
Thank you, Dr. Panchal and Kiran Bhai. આપણે જોયું બધાએ કે પંચાલ સાહેબે અને રાજપૂત સાહેબે ખૂબ સરસ રીતે માઇક્રોબિયલ ન્યુટ્રિશનનું પ્રેઝન્ટેશન કર્યું. હવે અહીંયા થોડાક પોઈન્ટ્સ હું આપને જણાવવા માગીશ જે થોડા ઇન્ટરેસ્ટિંગ પણ છે કે ઓર્ગેનિઝમ્સ જે છે એ સર્વત્ર બધી જગ્યાએ વાપેલા છે. હવા, પાણી, ખોરાક, આપણી બોડીમાં, બોડીની સરફેસ ઉપર, everywhere there is a presence of microorganisms. To that extent that certain groups of organisms, they are able to get that nourishment even in a water which is as good as a distilled water. ઘણી વખત આપણે જોયું હશે કે જો પાણીનો ગ્લાસ જે પીવાલાયક પાણી છે, જે આમાં આપણે માનીએ છીએ કે ડિસ્ટિલ વોટર કે મિનરલ વોટર જેવું છે, એને તમે થોડા વખત માટે અનડિસ્ટર્બ રહેવા દો તો ઉપર એક પાતળી મેમ્બ્રેન જેવું થઈ જાય છે. તો એ મેમ્બ્રેન નોર્મલી અમુક બેક્ટેરિયાની બનેલી હોય છે. આ ખૂબ ધ્યાનથી જોશો તો જ તમને દેખાશે. આવા ઓર્ગેનિઝમ્સ એમની એ કેપેસિટી છે કે એ વોટરની અંદર કે જેમાં આપણે એમ માનીએ છીએ કે કોઈ ન્યુટ્રિશન નથી, છતાં પણ એમાંથી કંઈક મેળવી અને ગ્રો થાય છે. આવા ઓર્ગેનિઝમ્સને આપણે ઓલિગોટ્રોફ્સ કહીશું. ઓલિગો મીન્સ a very small quantity of nutrients are there, but still they are able to obtain it from them. Then there is another group of organisms which are predators also. You will be surprised, but there is a bacteria like Dallovibrio. એનું નામ છે ડેલોવિબ્રિયો કે જે પ્રિડેટર તરીકે કામ કરે છે અને એક બીજા બેક્ટેરિયાના સેલની અંદર જઈ મલ્ટીપ્લાય થાય છે, ગ્રો થાય છે અને એ સેલને મારે છે. તો પ્રિડેટર પ્રે રિલેશનશિપ ફક્ત હાયર ઓર્ગેનિઝમ્સમાં છે એવું નથી, એવું આ ટાઈપનું રિલેશન્સ માઇક્રો ઓર્ગેનિઝમ્સની અંદર પણ જોવા મળે છે. ત્રીજી વસ્તુ એક પ્રોમિનન્ટ માઇક્રોબાયોલોજિસ્ટને હું એ કોટ કરવા માગીશ. Dr. Atlas, who was a great soil microbiologist also, and according to him, there is a microbial principle of or a principle of microbial infidelity. એમના મંતવ્ય મુજબ what he said was that there is no substance in a world which microorganisms cannot use that as their cellular constituent for their cellular constituents and therefore energy. It means એવું કોઈ પણ સબસ્ટન્સ નથી કે જે બેક્ટેરિયા ન વાપરી શકે provided you give them certain time and certain period for their growth. Like you must have come across certain names like Pseudomonas. Pseudomonas organisms અને એવા બીજા બે ત્રણ organisms કે જેની અંદર પ્લાઝમિડ્સ આવેલા છે. એ પ્લાઝમિડ્સની ઉપર જીન્સ હોય છે અને એ જીન્સ લગભગ એવા સબસ્ટન્સીસમાંથી એનર્જી અને ન્યુટ્રિશન મેળવે છે કે જે આપણે કલ્પના બહાર હોય. ફોર એક્ઝામ્પલ પેટ્રોલ ઓર certain disinfectants or xylene or certain aromatic compounds which are basically harmful for other organisms, toxic to other organisms. જો ઓર્ગેનિઝમ્સને આની અંદર એડપ્ટ કરવાનો સમય મળે તો એ પોતે એવી એન્ઝાઇમ સિસ્ટમ ડેવલપ કરે છે જેથી કરીને આ પ્રકારના ઓર્ગેનિઝમ્સ પણ ત્યાં આગળ ગ્રો થઈ શકે છે. આ સિવાય બીજી એક વસ્તુ કહેવા માગીશ કે જ્યારે આપણે ન્યુટ્રિશન વ્યવસ્થિત રીતે જાણી લઈએ કે આ બેક્ટેરિયાને આ પ્રકારની ન્યુટ્રિશન જોઈએ છે, that they require carbon, nitrogen, this and this, either in organic or inorganic form. The next step is basically on the basis of this knowledge we can formulate a medium. And when we can formulate a medium, it is possible to grow them in the laboratory. So medium is nothing else but it is a balanced formulation of nutrients required by microorganisms. ઇન્ડાયરેક્ટલી આપણે કહીએ કે સપોઝ આપણે જ્યારે જમવા બેસીએ છીએ ત્યારે જેટલા પ્રમાણમાં કાર્બન અને નાઇટ્રોજન સોર્સ, જે તે ફોર એક્ઝામ્પલ રોટલી અથવા તો દાળ જે પ્રોટીનનો સોર્સ છે, રોટલી જે કાર્બોહાઈડ્રેટનો સોર્સ છે, એટલા જ પ્રમાણમાં આપણે મીઠું નથી ખાતા. કારણ કે મીઠાની રિક્વાયરમેન્ટ્સ જેમાં સોડિયમ સોલ્ટ્સ હોય છે કે ક્લોરાઇડ્સ હોય છે એ ખૂબ ઓછા પ્રમાણમાં જરૂર છે, માટે આપણે એને શાકમાં અથવા તો રોટલીમાં ટેસ્ટ તરીકે નાખીએ છીએ. જ્યારે કાર્બોહાઈડ્રેટ્સ અને પ્રોટીન્સની રિક્વાયરમેન્ટ્સ વધારે છે તો આપણે એ ક્વોન્ટિટી આપણા થાળીમાં વધારે હોય છે. સો વેન યુ નો દેટ વિચ કોમ્પોનન્ટ્સ આર રિક્વાયર્ડ ઇન વિચ પ્રપોર્શન, this is how a formulation of a medium takes place. And when we are able to successfully formulate a medium, then we are able to grow them in the laboratory. However, it is also a very true fact that whatever microorganisms at present which we know or which the world knows, only say 10% of that microorganisms are known and very few of them they are cultivable in the laboratory. Still, by and large, there are so many organisms which are present surrounding to us either in soil or in sewage or in other environments where we have not been able to successfully grow them in the laboratory. Why? The reason is we do not know about their nutritional requirements. Therefore, the world of microorganism is very large, and to know that in detail it requires a lots of time. Still, we have been able to know only little amount of organisms, little bit of organisms about it, and then if we are able to know more and more in detail about the nutritional requirement, we will be able to cultivate more and more of them. પંચાલ સાહેબ, આપને કંઈક છોકરાઓને જણાવવું હોય તો 2 મિનિટ આપણી પાસે છે હજી.
આભાર રાકેશભાઈ. જ્યારે રાકેશભાઈએ કહ્યું કે સુડોમોનાસ પ્યુટિડા excellent bacteria છે. It's an organism which can even degrade the oil. So I would like to focus a prime requirement in case of all kind of accidents in environment, where few months you have seen that there was a oil spill, large oil spill in ocean near Bombay, and people are worried about that what would happen to the aquatic life. So there we do have microbes, and the microbes are the only remedy in this kind of accidents. Where the Pseudomonas putida, which is explained by Rakesh Bhai here, that is the organism. If you spray this organism and tactfully handle them, then these are the organisms which can degrade all this oil and at a faster rate, where no other method is employed. The microbes are at your remedy. Similarly, if there is a kind of radiation leakage, you may imagine what would happen when radiation leakage used to be there. So there also we have a kind of bacteria which is Staphylococcus radiodurans. These are the excellent cocci which has got capacity to absorb the radiation and can store radiation inside the cell. A large project, which is a kind of mega project on the research work on Staphylococcus radiodurans, is going on at MS University, Baroda, along with the association to BARC, Baba Atomic Research, Bombay. So there they work out this kind of bacteria. So any kind of problem comes to environment, the microbes are at remedy.
થેન્ક યુ પંચાલ સાહેબ, આપે બહુ સરસ જણાવ્યું કે માઇક્રો ઓર્ગેનિઝમ્સ ન્યુટ્રિશનલ રિક્વાયરમેન્ટ જાણવાથી આપણે પર્યાવરણને પણ થોડો બચાવી શકીએ છીએ, કે જેની અંદર ઓર્ગેનિઝમ્સ આવા જ્યારે ડિઝાસ્ટર થાય ત્યારે એનો યુઝ એમની ન્યુટ્રિશનલ રિક્વાયરમેન્ટ જાણીને આપણે કરી શકીએ છીએ. તો હું અત્યારે બંને સ્પીકર્સ જે મારી સાથે છે, ડૉક્ટર પંચાલ